You can, that's working. All right, what happened to this code? Okay, we will also do 771. Okay, so real quick in terms of updates, we have what happened to the rest of my flock. All right, good to know. So you have an exam, you have your lab exam next week. So the Tuesday section will be taking it Tuesday, the Thursday section will be taking it on Thursday. And we will be doing the sensory lab. So please look through and read that lab before you come. We'll be doing the sensory lab after the exam. The exam will be on Canvas. The exam will be on Canvas. You do not need a Scantron. You do not need Scantrons. You don't have to worry about it, but please make sure you bring some kind of device that's compatible with Canvas and locked up browser. Are we clear? Mm-hmm. So please make sure that it is a compatible device. So you're going to be doing that and the sensory lab, and then of course we have lecture. So lecture exam will be the week after that. Lecture exam is week after that. You have Monday off, not that it's relevant to us, but you have Monday off. So if you have other classes here, don't show up, okay? Monday is off, the campus is closed. So spend that time studying for the lab exam, play catch up. I will try and record and upload a review for the lab exam, okay? And I think the tutor is also doing those kinds of lab reviews every week. So have you accessed his lecture review? Yes, and is it, I haven't watched it, that's why I'm asking you, is it pretty congruent with what I'm doing? It may not be as detailed as mine, but it's a review, right? So I will see if I can do a lab review for the lab exam, because it is a lot of material. So that will happen. Again, once the summer anatomy schedule is made known, I'll let you know. I also told today's class that if you would like, I can make the, so I taught anatomy last semester. I used online videos as lecture, so it was asynchronous. So if anybody wants access to that, just email me, okay? And I'll send you those links, and I'll enable download, so you'll be able to download those videos and keep them for future reference if you want to use them. Okay? So they're not the best lectures, okay? Because I'm not an anatomy person, but looking at the way it was taught before, who knows, if the way I teach appeals to you, it might help you, right? So use it for what it's worth or dump it, doesn't matter. Just let me know, okay? Send me an email, I'll compile that link, and maybe another easier way is to compile that link and then paste it into a page on Canvas modules for your course and leave it there. So that might be even easier, right? So if you decide you want it, that way I'm not getting flooded with emails. So forget the emails. I'll compile it and I'll post it on Canvas, maybe in announcements or maybe in one of the modules. That's it. Any other questions? Any questions on what I presented last time? We started talking about neurotransmitters, and I told you that you can have excitatory neurotransmitters and inhibitory neurotransmitters, okay? And we're going to see how, in general, neurotransmitters work. Okay, before we go into excitatory, inhibitory details, because for that we have to talk about summation. So I want to talk about how neurotransmitters work, and then we've already talked a little bit about neuron anatomy, and I told you that graded potentials happen in the cell body, and then those graded potentials can get added at the axon hillock, and the axon hillock is where you find voltage-gated sodium channels first. And so action potential gets initiated at the axon hillock, and then it gets propagated, okay? So throughout the axon, we're going to look at that in detail also. And then since neurotransmitters primarily work on the cell body, I want to do neurotransmitter action here and wrap cell body off. Make sense? And then we'll move on to the axon, and then we'll look at what happens in the axon terminal. I'm hoping to get through all that today. I was hoping to start the brain, but I don't know if that'll happen. But this is a very important mechanism. You're going to be seeing this neurotransmitter mechanism over and over and over and over ad nauseum till the end of the semester. So again, this is all fundamental stuff you need to know, okay? All right, so in terms of, oh, one more quick thing, based on one of the questions I got in lab on Tuesday, actually. So the notes that you got from 103A, remember that document I uploaded for histology? You don't need to know 90% of it, by the way, okay? Because it's for 103A. Ignore all the anatomy, are we clear? I'm not going to ask you landmarks. I'm not going to ask you planes. I'm not going to ask you serous membranes. I'm not going to ask you any of that stuff, are we clear?
Yes.
So just ignore all that. Read the microscopy. That is relevant for you. Microscopy is relevant. So from those notes, I need you to only focus on two things: microscopy and all the histology, the 18 different slides, and you need to know what those slides look like and what the function is. That's it. I don't want you going into any more detail on that. Are we clear?
Yes.
Know the slide, know the function, the end. If you're taking anatomy simultaneously with this class, that document will be a godsend. So use it. Okay, it'll be very useful for you. Okay, because it's very detailed. All right. So neurotransmitter action. Okay, so here we go. NT is neurotransmitter level. So we're looking at neurotransmitter action. So neurotransmitters bind specific protein, and these are IMPs, okay, obviously because they're receptors. So they bind specific protein receptors, okay, and they will turn them on, and the scientific term is activate. So receptors will be activated. There's no situation that I know of where receptors are inactivated. That's dumb. Why bring a message? Okay. Now here's the interesting part. We've look at the neurotransmitters here. This is a thinking moment. Put your thinking cap on. You have excitatory and inhibitory, right?
Yes.
Yes. So shouldn't neurotransmitters also, in terms of its action, be able to activate and deactivate certain pathways, correct? Now what happens is the receptors always get turned on. Okay, they always get turned on. But the receptor can depolarize and hyperpolarize down the road. Am I making sense? Mm-hmm. I'll give you an example. If the receptor turns on protein A turns on protein B turns on protein C, signal transduction, right? So you go through this, and protein C turns on potassium channels. If you open potassium channels, what will you do? Hyperpole or dipole? Dipole. You're opening potassium channels. Think before you answer. You're not processing. Hyperpole. You're going to turn. So the neurotransmitter, even though it activated the receptor, eventually leads to an off signal. Am I making sense? Mm-hmm. Yes. So keep that in mind. So you can you turn the receptor on. This is guaranteed on your exam. I will ask it. Receptors will turn on, right? But the subsequent signal transduction can be depolarizing or hyperpolarizing. So you have the regulation there. Am I making sense? Yes. So I'm going to write that in disappearing ink. So subsequent signal transduction. This becomes very, very important. It becomes extremely important because particularly in the heart, you have pathways that turn the heart on and then pathways that turn the heart off. Yes. And they're both equally important. Okay? So subsequent signal transduction can lead to dipole, which is an excitatory moment, or hyperpole. which is an inhibitory moment. I'm going to let you absorb that concept and ask me questions. All right, looks like you're clear. So what are the two modes of action? So you can have direct acting or direct action. So direct action by definition is when a neurotransmitter binds its receptor, and I'm going to abbreviate that NTR, neurotransmitter receptor, NTR, okay? It binds its receptor, and NTR happens to be an ion channel. Now tell me, is this a guaranteed depolarization? Is this a guaranteed depolarization?
No.
Okay, because I said ion channel, right? What if it's a potassium channel? What if it's a chloride channel, right? Make sense?
Mm-hmm.
If it's a sodium channel, then yes, depolarization will happen, right? But if it happens to be a potassium channel, you'll hyperpolarize, right? So very important. So neurotransmitter binds its receptor. Neurotransmitter happens to be an ion channel. In other words, what happens is the binding of the neurotransmitter to the neurotransmitter receptor changes the conformation, which is a fancy way of saying what? It's a shape change, right? So what do you think shape change, how is that relevant to ion channels? Shape change, how is shape change relevant to ion channels? Open or close them, right? Exactly. Gating, right? Opening or closing. So binding of neurotransmitter to neurotransmitter receptor changes the conformation. Most likely, in this case, remember I told you it's always activation, right?
Mm-hmm.
So you're opening ion channels. Okay. If ion channel is a sodium channel, okay, you have sodium influx, which leads to, that's right, depolarization. Okay, this kind of channel is called, is called chemically gated channels. Correct? Binding of a chemical opens the gates, right? Chemically gated channel. Not difficult, okay? Does everyone understand chemically gated channel?
Yes.
It is also called ligand gated channel. They both mean exactly the same thing. Ligand gated channel or chemically gated channel, right? Ligands are chemicals. Okay, same.
Ligan?
Ligand. Ligand gated channels. Most cell biologists call them chemically gated channels. Most neurologists call them ligand gated channels. Okay? So, all right. If the ion channel, if the channel is potassium or chloride, okay, you will have hyperpole. Okay. Now here's $1 million question. The equilibrium potential, and this will tell me if you're thinking or you're just answering. Very tough question. So, if the equilibrium potential of chloride is -70, not if, it is, okay, it's experimentally determined. The equilibrium potential of chloride ions is -70, correct?
Mm-hmm.
What is the point of opening chloride channels? Why did evolution do this? Now tell me, what is the definition of equilibrium potential? The ion stops moving, correct? So if at -70, I open my chloride channel, right? And you're at -70 at rest, will chloride move?
No.
then why bother? What's the point of all this? Where is the mistake in my thinking? I used to ask these kinds of questions 10 years ago. Where's the mistake? What is the flaw in my analysis? Very, very straightforward. Okay, but a fact that you rarely think about and you should be thinking about. True or false? All cells have the same resting potential. False. Right? -70 is generic. Correct? Chloride, for example, in the eyes, if you open if you open chloride channels, it's going to rush in. Okay? Because the inside is -30. Am I making sense? It's not -70. It will go. It'll go slowly, but it'll go. Am I making sense? So
Yes.
don't always assume that everything is -70. That's what will happen when you memorize, when you blind memorize, and you're not thinking. Okay? So remember that being a chloride channel is physiologically relevant because tissues have different resting potentials. Many of them are -70. Okay? In most cases, it is relevant. And in those cases, chloride channels are minimally expressed, right? But also understand that chloride has a concentration gradient. Chloride is high outside, low inside. Clear? So that will motivate some chloride to go in, okay? So you're going to get hyperpolarization because you're gaining negative charge. Clear?
Yes.
All right. So that's direct acting. Again, if you look in the PowerPoint, I have pictures for all of this. So take a look at those pictures, okay? Now tell me, will direct acting be fast or slow? All you have to do is open a gate. Can you do that fast or is it going to be slow? Compared to, let me give you a comparison here. Compared to, so let's say that I'm asking the people in the last row to open the doors versus I'm whispering it, right, to people here. They whisper it to the next row. They whisper it to the next row. Are you seeing the signal transduction here?
Yeah.
So now tell me, is direct acting slow or fast?
Fast.
It's rapid, okay? So absolutely rapid. Because signal transduction has many, many steps. And when you have many steps, you're going to be
Slow.
slow. What have you learned so far that is an example of that? Where you have multiple steps, so you're going to be slow. The working of a
Neuron. Okay.
not that general. Working of a neuron, several fold, right? It does many things. Let me give you a hint. It's inside us, it's part of a cell. Active transport, sodium potassium pump, several steps, right? Therefore it's very slow. Remember, all primary active transport is slow. Remember, why did I say it was slow? Too many steps.
Yeah.
Now do you see how to connect concepts?
Yes.
Yes? Okay, so so you have this, so this would be hyperpolarization, so I'm going to highlight that. Okay, and so direct acting is very rapid. It's a rapid response. Okay, and there's no need for signal transduction because the receptor itself is an ion channel. Are we clear?
Yes.
When I present to you indirect acting, you'll understand this a lot better. So indirect acting, majority of neurotransmitters are indirect acting. Please make a note of that. Majority of neurotransmitters are indirect acting. Majority of NT, okay? And signal transduction is required. And I'll post these notes today. Okay, so signal transduction is required for indirect action. Okay, and here's the way it works. So neurotransmitter binds neurotransmitter receptor. Neurotransmitter receptor activates protein 1, which then activates protein 2, which then activates protein 3, blah, 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 blah. Okay, and eventually you will activate an ion channel. Now tell me, is this going to be slow or fast?
Slow.
Slow.
Very slow, okay? So this is going to be very slow. Now, you want the indirect acting to be slow because it deals with a lot of very important life controlling decisions. Okay? You want them to be slow. You don't want to rush into these type of responses. Okay? Direct acting is really rapid. I'll give you an example. Skeletal muscles are direct acting receptors. Okay, you'll learn this next chapter. You just saw a line. You want to do signal transduction and wait till your muscle starts working.
No.
You want to jump up and run, correct?
Mm-hmm.
You don't have time, right? So evolution picked direct acting. So whichever foolish species had indirect acting for skeletal muscle got weeded out real fast, right? Because they were preyed on.
Mm-hmm.
Okay? So skeletal muscle activation needs to be instant. And you know that, right? Brain is thinking while my tongue is talking. Can it get any more instant than that?
No.
Yeah. Okay? So indirect acting is not like that. Indirect acting, a lot of these are very high energy requiring and for example, increasing heart rate. Do you want to increase heart rate immediately? Your blood vessels will burst. Am I making sense? You don't want that, right? You need a certain amount of slow to it, and the heart itself will burst. Are we clear?
Mm-hmm.
You don't want that. So majority of neurotransmitters are indirect acting, and they're slow. Relatively slow compared to direct. Direct is very few, far in between, and fast. 3 Fs. Few, far in between, and fast. Okay? Any questions on direct acting versus indirect acting and what that means? Did everyone say hi to our new visitor? Say hi. Welcome to our class. Is this too boring for you? Me too. All right. So, any questions on these two mechanisms? Okay. In nursing school, you will learn specific neurotransmitters. And if you take 103A, you will learn specific neurotransmitters. I'll make you memorize those, memorize that table. But you're not. You're in 112. I'm not going to waste your time. You can learn that in nursing school. You don't need that here. I'm not going to make you memorize those. The only 3 neurotransmitters I need you to know are going to be epinephrine, also called adrenaline, noradrenaline, so that's 1. Together they're 1. And actually, I guess you could count them as 2. So those are 2, and then acetylcholine. Acetylcholine, okay? And that's parasympathetic. So you need to know these 3. This is all we'll look at in each of the chapters. Acetylcholine is it's a class of its own. It does a variety of things, okay? But you're going to hear acetylcholine, adrenaline, and noradrenaline a lot. Those are the 3 neurotransmitters you need to know. Acetylcholine can be direct or indirect, okay? Epinephrine or adrenaline is never direct. It's always indirect. Are we clear? Because you're turning on the fire alarm in your body. You do not want that to be rapid. Okay? So adrenaline, noradrenaline are both always indirect acting. Always. There's no exceptions. Everybody got this? Okay.
Yeah.
All right. So let's look at, so that's direct acting, indirect acting for you. Why this is fast, why this is slow. When we do hormones, we're going to go over this pathway in detail, right? It's called the amplified pathway. So we're going to be going over that in detail there. And we're also going to look at it when we do smooth muscle. We're going to be looking at that pathway there. So I'm not going any further down. I just need you to know the general definition of direct acting and indirect acting. Everybody got this?
Yeah.
Okay. So this is part of what happens on the cell body. So the last event that happens here is summation, and very important concept. So when we're doing summation, it's a fancy word for adding. Okay, summation is a fancy word for the word addition. You're literally adding all the voltages, right? And I will tell you what this means with an example. So first understand the neural circuit. So if you take a neural circuit, it looks like this, right? So the neural circuit basically consists of one neuron connecting to many other neurons. Yes?
Mm-hmm.
Yes.
Okay. And it's just like if you have LinkedIn or if you've seen LinkedIn, it's just like LinkedIn, right? I am connecting to several of you, right? Right?
Yes.
And several people are connecting to me.
Yes.
Do you understand what I'm saying? It's one big
Yes.
tree, right? So that's how neurons are, okay? So you're getting inputs from a lot and you're giving inputs to a lot, right? And this is in the brain, the average in the cerebral cortex is 150,000 synapses. 150,000, right? It's a lot of neurons connecting with each other. So the meeting point of two neurons is called a synapse. And the word synapsis means bring together, okay, literally. So synapse is where two neurons, so the gap between two neurons, okay, it's called a synapse. Now, there are three kinds of synapses, that's all anatomy, I'm not going into that. But a synapse is basically a gap between two neurons. And I'm saying it's a gap because we're talking about humans. When you look at amphibians and fish, it's actually an electrical synapse where they're physically connected to each other, right? Whereas if you look at humans and more advanced vertebrates, quote unquote advanced vertebrates, it's more an electrochemical synapse. That's why it's a gap. It's a little space, okay? So what happens is the following. So this neuron is called the presynaptic neuron. And the definition of it is that it secretes neurotransmitters. So the source of the neurotransmitter is your presynaptic neuron. Pre means before the synapse, correct?
Yes.
And so these, therefore, would be called postsynaptic neurons. Do you agree?
Mm-hmm.
Okay, these are postsynaptic. And I would note this down. Presynaptic is always a neuron, no exceptions. Presynaptic has to be a neuron, by definition. You're releasing neurotransmitters. If you're releasing neurotransmitters, what type of cell are you?
Neuron.
A neuron. Okay, so note that down. So very important. Presynaptic cells are always, they have to be neurons. Okay. Postsynaptic, not really. Okay. So postsynaptic could be neurons. It could be glands. It could be muscles like erector pili. We went over that before, right? Thermoregulation, erector pili. So it could be a muscle like that. It could be blood vessel muscles, remember? Right? It could be skeletal muscles, right? That receive nerves. Anything that a nerve goes into, right? It could be glands. It could be other neurons. It could be exocrine glands, endocrine glands. It could be muscle tissue, okay? Am I making sense? Postsynaptic is varied. Presynaptic has to be a neuron. Is this clear?
Yes.
Okay. Postsynaptic receives the neurotransmitter. So here's a thinking question again. Where would you find the neurotransmitter receptor? Where would you find the neurotransmitter receptor in this entire diagram? Let's see how specific you get. Mark?
The dendrites?
Yeah, I think that's as specific as it gets. So you will find dendrites, no, not specific as it gets. Bad, Mark. Bad me. You're 99% there. Dendrites of what? My grandmother? Dendrites of what? Which one? Yeah, dendrites of the postsynaptic neuron. Am I making sense? Are you learning to be specific? Learning, let me reduce that. All right, so neurotransmitter receptors, I'll write that out. So neurotransmitter receptors are localized, remember, Queen of England, localized to the cell bodies, and not just dendrites, Mark, okay, but mostly dendrites, but to the cell bodies of postsynaptic neurons. Are we clear?
Yes.
So where will you find the response? Presynaptic or postsynaptic? Response to a neurotransmitter is found A, presynaptic, B, postsynaptic, C, both, D, neither. Where will you find the response to a neurotransmitter?
Postsynaptic.
Only postsynaptic, right? Why would presynaptic respond?
Yeah.
Correct?
Yes.
So cell bodies of postsynaptic cells. I'm going to leave it as cells. Remember, postsynaptic cells can be neurons. Okay. This can be neurons, right? I mean, if these are neurons and you have cell bodies, but postsynaptic could be muscle. Okay, it could be glands. Okay, etc. as well. And the phenomenon, so this has a special name. This is called innervation. I'll be using this a lot. Okay, just like vascularity, this is called innervation. And again, anatomy should have covered this. Innervation means going into some location. Nerve going into a location is called innervation. In, going in, right? Any questions on this?
That means the receptors do not need to be on a dendrite of a cell?
It mostly are on the dendrites,
Okay.
but it could be pretty much anywhere on the cell body. In fact, some receptors are localized on the axon hillock also. Okay. Now, I'm going to, so you understand what excitatory means and what inhibitory means, right? Excitatory means you depolarize. Inhibitory means what? You hyperpolarize. Now, I don't care how you act. You could be direct acting or indirect acting. Right now, we're talking about whether you're excitatory or inhibitory. Are we clear? I don't care which mechanism. Is this making sense?
Mm-hmm.
Okay. A lot of times people mix these up, right? They memorize direct acting to be excitatory only, and indirect, because people tend to do this, right? When your learning is not deep enough, you tend to mix these up. Don't connect things that don't belong. Do not connect things that do not belong, right? So there's no need to, keep them separate. So again, I repeat, excitatory means you have the possibility of an action potential, right? Inhibitory means no chance, right? You're hyperpolarizing. Okay, so back to summation. So if you take a postsynaptic membrane, I don't care what this is, but we're going to assume this is a neuron for simplicity's sake. So I'm going to say this is a postsynaptic neuron. It can have a variety of neurotransmitter receptors. Do you agree?
Mm-hmm.
Okay. So I'm going to say that I'm going to call this NTR1. Okay. And I'm going to call this NTR2. And I'm going to call this NTR3. Okay.
Yes.
It's just a name, okay? NTR1, 2, and 3. And I'm going to tell you, and I'm telling you this, that NTR1, which is highlighted in green, is excitatory. So that is excitatory. Okay, and if you look at this orange, is going to be inhibitory. So far, so good?
Mm-hmm.
Okay, so, oops. Okay. Okay, that's inhibitory. And this
The
is,
highlights look the same color.
I'm sorry? Oh, wonderful. I promise you it'll look different on your notes. It looks very different here. So, let me do it this way then, okay? So this is excitatory, this is inhibitory, okay? So here is inhibitory. Does that at least look different? Okay, good. And then these two are excitatory, okay? Can two different neurotransmitters be excitatory? Can two different neurotransmitters be, this is not a thinking question. Can two different neurotransmitters be excitatory?
Yes.
Yes, of course. Okay. So what does this mean? This means that this will increase voltage, correct? Okay. This will also increase voltage. And this one here, okay, will decrease the voltage, correct? Now, if all this adds up to threshold, listen to my question. If all three add up to threshold, will you have an action potential or not?
Yes.
Yes, it's all or none, right? Yeah. Signal will go through. If these three don't, okay, that means the signal is cut. Does that make sense?
Yes.
So, do you see how I've added the voltage? This is called summation. Now, there's only one caveat in this system. Can there be a time lapse between NTR 1, 2, and 3?
Yes.
Think before you answer. If there is a time lapse, don't you think the sodium potassium pump would already correct it?
Oh, that's what
you cannot have a time lapse here. Okay? All this is assuming. So when can you add the voltages? When is that relevant? When all three neurotransmitters simultaneously activate their receptors. Are we clear? So there is a simultaneousness to this. Simultaneity? What is the noun of that? I have to find out. Queen Victoria in me tells me it's not simultaneousness. Okay. This particular one, right? Everybody see this? So you've got three different neurotransmitters producing changes in voltage. If these voltages add up to negative 55, or in my example, negative 63, you will have an action potential. And guess where they're getting added? At the hillock. Am I making sense? So the cell body receives it. Yes? Cell body receives it. This kind of reminds me of, the whole diagram reminds me of Mr. Mackey from South Park. Because that's what his head is like. It's cell body, and then his neck is the axon hillock. It's that thin, right? He looks like a neuron, right? The rest of the body is an axon, right? Really. Anyway, I shouldn't say South Park. We have a child. So, la 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 la. Okay, here we go. So the summation happens in the cell body, and the consequence is judged at the hillock. The voltage is judged at the hillock. So think about it. So let's say that I'm going to give you some example values. These are not real values. These are example values. Okay? Are you with me? So example. So let's say that NTR1 is it produces plus 20 millivolts, right? And NTR2 produces let's say negative, negative 20 millivolts, right? Okay, and NTR3, okay, produces, somewhere about another negative 20 millivolts, okay? That can't, because it's excitatory, so not negative 20. So let's say that it produces something else. So let's say that NTR3, so I need negative 55, right? So let's say that this produces 15 millivolts of a response, correct? 15 millivolts. So when you add these up, what do you get? So the plus 20 and the minus 20 will cancel each other, correct? 15 millivolts, when you subtract from negative 70, 15 is what? Remember, I only want negative 63, right? 15 is negative 55, right? Is that higher than negative 63?
Yes.
Math,
Yes.
yes, right? So will you have an action potential?
Mm-hmm.
You will. Now, what if all three were plus 20? Or, let me ask it this way. Let's say NTR2 is out of question. It's not there, right? That's a very strong signal. Are you seeing that?
Mm-hmm.
But because NTR2 is there, it becomes a weaker signal. Does it matter for action potentials?
No.
No, because it's all or none. This is where we have imperfection in the system. All hell evolution. That is pathetic creation. Don't you agree? This is not creation. Somebody in 103A asked me a question and I said, that's because I created it that way. And they didn't get it at all. I wanted to fail them all. So anyway, and then I had to explain that joke and it died horribly. So the point is, then why would you evolve something like this? Why do you have inhibitory neurotransmitters? Because it's not consequential. The consequence is this. Yes, I agree that whether you have minus 55 or whether you have minus 53 or whether you have minus 61, you're going to have an action potential because you're higher than threshold, all or none. But the frequency of the action potential is quite different. Do you understand what I'm saying?
Yes.
That depends on this initial stimulus. If this NTR2 is not there, you're going to be firing like heck to the brain. Am I making sense?
Mm-hmm.
If this is there, you're firing a lot more slowly, right? So if I'm doing this versus, right?
Mm-hmm.
The effect on your brain is significantly different because how you interpret everything is by what? What is the basis for interpretation of stimuli by the brain? How does the brain interpret stimuli? Frequency of action potentials. So this will make a difference. Or am I making sense?
Mm-hmm.
Muy importante, okay? So, so remember that inhibitory signals are extremely crucial. Inhibitory signals can very much modulate the frequency of the resulting action potentials and therefore how the brain interprets these signals. Are we clear?
Yes.
For example, in the eye, if an inhibitory signal is not there, right, and you send a very strong signal, you will perceive, your brain will think something is green instead of it being orange. Make sense?
Mm-hmm.
So it can result in significant mistakes. And so people like that, or animals like that, evolution will make you extinct. Are we clear?
Mm-hmm.
So these are probably, everything is trial and error. Everything is trial and error. Okay? Any questions on this kind of summation? Beautiful process. Beautiful. Isn't my creation something? Anyway, also lost. But it's because of time. 3:30, I'm not expecting anybody to laugh. Okay, I'm happy your eyes are up. So, I'll tell you how cruel I was once. And in high school, this is when I was younger, so of course we're talking BC, Ice Age. And I was teaching the same class actually, anatomy and physiology. I was teaching at a magnet school, so I taught A&P with the same textbook to high schoolers. And I had a student, this was 8 o'clock in the morning class, and always nodding off, okay? Always nodding off. So I would make him sit like this the whole class. I'm not kidding, okay? I do something like this nowadays, you'll meet me in San Quentin. Okay, so I would be like, no, Tynan, open your, he's not a cardiologist, but I'm like, open now. Okay, and he would sit like that the whole class. Okay, and then I used to think about, my God, of course, those kids loved me, and I loved that. So I was able to do things like this stuff for them to pay attention. They know that I wanted them to learn. And that was the thing behind this, and they all did really well, too. So, and we dissected cats instead of cadavers, but yeah, it worked. I know I was somewhere, and I'm lost. So is this summation making sense?
Yes.
Okay. This type of summation, question.
When you get the chance, can you write down
significance of the inhibitory signal, okay, I'll write it up. Shouldn't be hard. I'm trying to conserve ATPs. No, I'm not. I just forgot to write it. Okay, here we go. So, inhibitory signals, this is an important point. Inhibitory signals modulate the frequency of subsequent action potentials sent to the brain, okay? Which will affect how the brain interprets the stimulus. Does that make sense?
Mm-hmm.
This type of summation, where you have simultaneous neurotransmitter feed into the postsynaptic, okay? So simultaneous effects of different neurotransmitters, okay? This is called spatial summation. Spatial summation. So think about spatial summation. It makes sense that spatial summation has excitatory and inhibitory, correct? Because you're adding a variety of signals, right?
Mm-hmm.
For spatial summation to occur, true or false, for spatial summation to occur, you can have only one presynaptic neuron. I repeat, true or false. For spatial summation to occur, you can have only one presynaptic neuron. Here's the hint.
Can't the same presynaptic neuron secrete different neurons?
There's the
entities?
key question. Can one neuron secrete multiple neurotransmitters? As far as I know, no. They're very specialized. Now answer my question.
Only one? No.
So a neuron can only secrete one neurotransmitter. Now going back to my question, spatial summation requires that there only be one presynaptic neuron, true or false?
False.
False. You must have multiple neurons. If I'm a neuron and I secrete only one does one neurotransmitter add spatially? No. Am I making sense?
Yes.
Okay. So it can add, but in a very different way. Okay, we're going to see that second mode now. But not spatially. Spatially requires different, look at, this is why you have to be anal about definitions. You need different neurotransmitters, are we clear?
Mm-hmm.
For spatial to happen, and it has to be simultaneous. What if I said you release one neurotransmitter, 10 seconds later you release another, and 10 seconds later you release another? That won't work. It has to all come at the same time. Is this clear?
Yes.
Okay. So you do have a situation where there is only one neurotransmitter involved, and there, okay, so that summation is called temporal summation. So temporal summation is said to happen, okay? So here, one neurotransmitter is released at a certain frequency. Is time involved?
Yes.
And what does temporal mean?
Time.
Time. Go by the name. Okay, at a certain frequency. So what does certain frequency mean? That's too ambiguous, right? If I release 10 neurotransmitters a second, I'm making this up. If I release 10 neurotransmitters a second, right, the voltage can hit negative 63. Anything less, I won't hit negative 63, right?
Yeah.
So if I release 10 neurotransmitters a second is when you get a signal. You get action potential. If I release 8 neurotransmitters a second, what will happen?
No threshold.
Nothing. You're at rest. Am I making sense?
Mm-hmm.
So true or false? True or false, you have an inhibitory neurotransmitter action when you release less than the critical frequency. Think hard, think definition. You have an inhibitory neurotransmitter action when you have less than ideal frequency.
False.
You're comparing apples to broccoli. Am I making sense? Inhibitory action by definition is what? Definition, inhibitory, hyperpolarization. If I don't release enough neurotransmitter and the membrane is still at rest, are you hyperpolarizing? No, so it's not inhibitory anymore. Does that make sense? Very, very important. Just because you don't have an action potential doesn't make it inhibitory. Are we clear?
Yes.
You cannot have action potential in the postsynaptic cell if, number one, you have actual hyperpolarization, right? That's one possibility. Or you have a temporally acting neurotransmitter that's not released sufficiently. But those are two different states, because in a temporally acting neurotransmitter, you don't hyperpolarize. You don't become negative 80. You're still at negative 70. Am I making sense? So that brings me to an essential point here. Temporal summation can never hyperpolarize. Does that make sense? You can't think about it. It's illogical. Why would you want to release more and more and more of a neurotransmitter to keep the postsynaptic cell from firing? It's already not firing. People, am I making sense? I can't scream more than this. My vocal cords will tear. And then you'll have to read my lips, okay, for a lecture.
And what's temporal summation for?
I'm coming.
Okay.
Patience, Patrice. So, I need to stand because my butt's asleep. Okay, everything else is asleep. Old age. Okay, so what is the point I'm trying to make? The point I'm trying to make is, so everybody think of yourselves as evolution. Why would you want, in your right mind, the room has no lights, correct?
Mm-hmm.
The room switches off, yes? If I go to the switch and press the off button till my fingers break, what would you call me? You will make me sit and do ECG with 20 electrodes around my head, correct? Make sense? Because that's nuts. The signal's already off. Who cares? Why do you want a stronger off signal when default is already off? Is that logically making sense?
Yes.
Yeah.
In spatial summation, it makes sense because you're turning on switches. So having off switches, it makes sense. Remember I don't know if you have to, but there are these annoying switches. I can't stand them because I always get them wrong and I can never master them. So there are these switches where there's a switch near the door that I have to turn on, and then I have to turn off the switch near the table for these lights to turn on. It's very annoying to me, right? Like, what is the point of this? Okay, I don't understand. Okay, but those are created by humans. Okay, so it explains the level of intelligence there. So the point is, you don't want a strong off signal. It makes no sense. So temporal summation is always excitatory. And where do you use it? Several places. Skeletal muscle. Imagine, I'm lifting this ruler. Yes?
Yes.
My brain will tell my hands, this ruler is really light. Yeah? You need maybe 2 fingers to lift it, right? I'm done. Okay? If I were to lift this table with that ruler, okay, and if my brain is saying, just use 2 fingers, right? Immediate EEG moment. We should put that whole machine in me, okay? So, because that's nuts, right? So when you have a huge box, right, and I'm asking you to lift it for me, right? You're gonna go, right? All the energy and getting all your muscles warmed up, and then you're gonna lift it, right? And fun is had when that box is empty, right? And what happens? You fall, right? This is what we did to our pledges after we had those accidents of bananas and water poisoning and all that. This is our extent of pledge hazing. We'll tell them to lift a box, and the box is empty, and they fall, and we all laugh, and then do other things. But the point is, that's called recruitment. Your brain is recruiting a lot of neurons because you're anticipating a heavy load. Yes? So there, a neuron can only secrete so many. You can only exocytose, which is bulk transport, which depends on calcium coming in. You can only exocytose so many at a time in terms of vesicles, right? So if you want to lift a really heavy box, what do you want to do? You want to crank up that neuron, okay, to release a total of at least 1,000 acetylcholine molecules, so your muscles get ready and you're ready to lift a heavy load. Am I making sense? Whereas if I'm only picking up a ruler, a plastic ruler, I'm probably only secreting 2 acetylcholine molecules. Does that make sense? And I'm recruiting only my fingers for that, right? Whereas for something like this, I'm going to recruit my whole body. Is this making sense? Yes. That's temporal summation. Everybody got that? Even more abstract example, learning. How many of you have heard of the word review? Bane of my existence. Right? Ooh, can I get 1,000 review videos that I'll never look at? So you could tear your vocal cords and drive yourself nuts. Sure. So what does review mean?
Review again.
Look at it again, right? When you look at it over and over and over, you're doing temporal summation of the brain. And when you do temporal summation, you make neurons, you don't need to know this, you make neurons more, oh, what the heck, know it. You make neurons more and more and more sensitive. Have you heard of the word use it or lose it? That's what it's referring to. The more you use your brain, and they're saying this now, right? You do puzzles or you do something intellectual in your older age, you slow down Alzheimer's, you slow down dementia, you slow down. That's because you're using your neurons. And when you use your neurons, they become more and more sensitive. Very easy. All they do is they make more neurotransmitter receptors. So when you learn, that's an example of temporal summation in the brain, in the hippocampal neurons. Does that make sense? That's how you remember. Okay? And when you do that, you're making these neurons more and more and more sensitive to many different neurotransmitters. Are we clear? And you're making those connections. So that's an example of temporal summation. Okay?
Yes.
Got this? So temporal summation can never be what?
Inhibitory.
Inhibitory. Okay, it can never hyperpolarize. I will ask questions on this. Okay, so examples of this would be skeletal muscle, recruitment, and this phenomenon called potentiation. Okay, and this is the basis of learning. Any questions on temporal summation and spatial summation? Okay, I need to review that again. So spatial and temporal summation, any questions? If I take a signal, okay, I keep repeating that signal, right? The cells respond by becoming more sensitive, okay, because you're using it a lot. Clear?
Yes.
Okay. And in some weird way, even muscle memory is an example of temporal summation, right? Your body automatically knows, right? So when you're getting to a particular turn, your body will automatically make the turn, right? And dancing and choreography and things like that. Those are all examples of temporal summation. Okay, most movement, okay? Anything else here? 5-minute break. When we come back, we will start with the brain. Okay, so we're done with this unit. So we're going to, for the rest of the period, we're going to do brain. And then next class, when we come in, and I'll do a little bit of spinal cord if I have time, otherwise I'm not going to bother. Next class, we will go into autonomic nervous system, which is really, really important. That you really need to know for physio. So we'll go deep into that next class. So I didn't even include brain in the syllabus because you cover that in anatomy. But if I don't give you that introduction, a lot of what I say won't make sense, right? There might be gaps. You're like, wait, what is that? What does the word nucleus mean? What does ganglia mean? I mean, all that is going to be confusing, right? So you kind of need, it's like putting a legend on a map. You kind of need to know what's what, right? So for that, you need some anatomy. So I guess, forgive me if this is too much anatomy here. But I'm gonna go over some really basic parts of the brain, and just again for context sake, for what's coming later. Do you need to know what I'm presenting? Yes, for the exam. These would be the easy questions, right? Because all you have to do is match the part to its function. That's it, right? Can't get easier. No thinking involved. Okay, so yay, here we go. So if you look at the brain, we are dealing with. Oh, thank you. Yes, speaking of brain, get one. That was the translation there. All right, here we go. Come on. I like that number. Okay, here we go. 6336. Yeah, okay. So when we look at the brain is covered by meninges, right? It's highly protected by 3 membranes. You don't need to know them. I'm not going into them, not important. We're looking at the functional biology of the brain here. So the cerebrum is the biggest part of the brain. So the main parts of the brain are going to be the following. So you've got the cerebrum, okay, which is in humans the biggest part of the brain, okay? And the cerebrum is divided into 2 hemispheres. Okay, so we have 2 cerebral hemispheres, right? And then we have the midbrain, okay, which is the middle portion, and it consists of some parts there. So we have what we call the corpora quadrigemina. We have the, although this is mostly in the hindbrain, I'm going to say some critical nuclei like basal nuclei. Okay, also called basal ganglia. So the word nuclei in the brain, the definition is different. It's a collection of cell bodies in the central nervous system. So again, this is all anatomy. So a nucleus is collection of cell bodies. Everybody knows what cell body is in a neuron, right? Collection of cell bodies in the central nervous system. When you say ganglia, it's a collection of cell bodies outside the central nervous system. Okay, so a nucleus is a collection of cell bodies inside CNS. A ganglion is collection of cell bodies outside the CNS. I will not ask this on my exam, therefore I will not write it. Okay, so you don't need that. But that's where this word comes from. Basal nuclei, they're very important. So they're found there and some other structures. Okay, not going into that. So hindbrain is crucial because here's where many of the important parts show up. The thalamus, okay, the hypothalamus, okay, to a small extent epithalamus, but I'm not talking about that. So the thalamus, the hypothalamus, these are all functionally really crucial. The medulla oblongata, pons, and the medulla oblongata. Okay, so all these comprise the hindbrain. Now, it's a rough classification. Okay, some people may say that midbrain has part of the thalamus. Probably, who cares, right? There are no set borders. Do you understand? I mean, we just divvy them up for our convenience. Okay, so these are all the important parts of the brain, and you do need to know what these all do. In addition, we also have a cerebellum, which means a tiny cerebrum. It's a smaller version of the cerebrum called the cerebellum. But believe me, it's worlds apart. It just looks like a smaller version. Worlds apart. Cerebrum is extremely complicated. Cerebellum is near dumb, right? The activities are very, very mundane. Okay, nothing complicated happens there, at least as far as we know. Okay, so these are all the different parts of the brain. So we're going to quickly look at how these are all organized, right? So if you look at the cerebrum, so cerebral organization, okay, I told you that you have two hemispheres. The two hemispheres, okay, are not identical functionally. Very important. They're not identical functionally. They look exactly the same, okay, but functionally they're not identical. Okay. For example, the left hemisphere houses what's called Broca's area. Okay, that's important for speech. Okay, the right doesn't. Are we clear? Left does, right doesn't. And so there is some uniqueness to these hemispheres, right? So this phenomenon, and I do need you to know this, this phenomenon is called lateralization. So lateralization is uniqueness of hemispheres, of hemispherical function. Uniques. Wow, we're really out of it. Hold on. Uniqueness, okay, of hemispherical function. Okay, that's called lateralization. Lateral means not being the same, taking sides, right? So lateralization. And if you look at the cerebral hemispheres, okay, so again, anatomy a little bit, bear with me. So this is called anterior, okay, or frontal, right? This is anterior, and this is posterior, right?
Yeah.
So as you go from anterior to posterior on the brain surface, okay, the brain surface, and in general, I do want to mention, this is important, external gray matter, okay, and internal white matter. So if you cut the brain, okay, you'll notice that the white matter is found inside, okay? Gray matter is cell bodies, white matter are all the axons. Remember myelin? Myelin's fat, right? What's the color of fat? White or yellow, right?
Yes.
Okay? So external gray matter, internal white matter. It's important. So keep that in mind. Now, cerebral organization, so understand that there is a very important marker, okay? And I can quickly draw this for you. So if this is the cerebrum, right? So there's a very important marker here called the central sulcus, okay? Okay, so this is your central sulcus. Sulcus means depression, yes? Like mine. So, I want to go to bed. Okay, so central sulcus. So there is a central depression there, and that's your landmark. It's really important. And this is, again, your face. This is your back, okay? Are we clear? Orientation, right? So, anteriorly, we have mostly motor areas. So anterior to the central sulcus is mostly motor areas, okay? And posterior, and motor is always written in red, default, okay? And posterior is always sensory. Okay? Functionally, this is important. Motor means these neurons begin in the brain and they will coordinate your movement. Motor means movement, right? Motor car, motorcycle, right?
Yes.
Movement. So they will initiate movement, but it's not just movement, it's any response, correct? So motor neurons originate in the cerebral cortex and reach target tissues. Are we clear?
Yes.
What about sensory? Sensory neurons bring messages to the brain, right?
Yes.
So motor would be efferent pathways, and sensory would be
Afferent.
afferent or afferent pathways. Are we clear?
Yes.
So I'll write all that down. So sensory are called afferent neurons. So they bring action potentials to the brain. Okay? They're also called ascending pathways. You rise up to the brain, correct?
Yes.
Going up to the brain, right? Ascending. Okay, so what would motor be? Afferent neurons, okay, that take action potential from the brain to targets, right?
Yes.
So you would call this
Descending,
right?
Mhm.
So to draw this, you can easily draw it like this. Okay, so that's a motor neuron. Okay. Oops. Blue. That would be a sensory neuron, correct?
Yeah.
Very important. And all this happens through the spinal cord. Clear?
Mhm.
Most of them, not all the time, most of them happen through the spinal cord. For example, neurons coming to your eye, they don't go through the spinal cord. They come right from the brain to the eyes. Okay, so is ascending, descending make sense?
Mhm.
Yes?
Yes.
Now, each body part is dedicated a particular region in the brain, okay?
Mm.
Now, the more you use that body part, the more area you get in the brain. The less you use that body part, the less area you get in the brain. For example, okay, your chin gets a very tiny area, right, compared to your cheeks.
Yes.
Because your cheeks are the basis of your facial expression, right?
Mm.
And cheeks also chew your food, right? And facial expression. They have a variety of skills, right? So any skilled area gets a greater portion of allocation of the neurons. Are we clear?
Yes.
So skilled areas. So if you look at the thigh, for example, compared to your face, there is no skill to the thigh, right? What does the thigh do? It bears your weight. That's it. Okay? Nobody says, I have a very skilled thigh, you know? Okay? There's nothing to do there. Okay? So, and by the same token, your fingers versus your toes, right? Your fingers help you write, right? I mean, you know, you can't normally do that with your toes, right? Your toes have limited movement because of the weight-bearing nature of your legs, right? So your fingers will have much greater number of neurons, both sensory and motor. Okay? Dedicated to them. Are we clear? So this is the principle of unequal representation. So if you think of this, very, very good analogy that I literally just thought of just now. Surprised my brain's working. The bigger the population, right?
Mm.
The bigger the population, the more representatives you're going to send to the Congress, correct?
Yes.
It's population based, correct?
Yeah.
And the smaller the population, the less representation. Are we clear?
Yes.
It's the same idea. Except here, it's not bigger the population, bigger your skill, right? The more skilled a muscle is, right? The more representation it's going to get. So I'll write that. So more skilled an area, here, God, area, more cortical representation, which means every body part does not get equal representation. Are we clear?
Mhm.
Doesn't. There are people who can write with their feet. It's actually more people than you know, people who are born without limbs, right?
Yes.
They learn to write with their feet. And I've had a student like that, actually. She would write with her feet, beautifully, actually.
Yeah.
And so what does that tell you? This is not set in stone. Can you learn?
Yes.
Yeah. If you're forced to, you can learn. Make sense?
Yes.
So that is called plasticity. Okay? So this is ability to change representation in the cerebral cortex. That's called plasticity. Now, when you're younger, the brain is not fully set, right? Yes? So you can make these kinds of changes fairly easily. This is why children learn so much easier.
Yeah.
Yes?
Yes.
As an undergrad, minimum 30 to 35 units a semester is what I took, right? And then grad school happened, very sad days. And then I joined Ohlone. I was 36, and I was bored out of my skull. So I said, you know what? I'm going to do a master's. So I went for my master's in education. You will not believe I could not even handle a single class. My brain had decayed so much by then with age, and 36 is not that old, but huge difference between what I could do when I was 19 to very humbling experience. Okay? I almost wanted to quit that program at least 80 times. Okay? I was like, this is not worth it. This is not worth it. I'm not doing this. And this is a subject I liked. Right? I wanted to learn how to teach. Okay? And I finally finished it. Okay? But I tell you, I kept cursing my age. I said, boy, does age take this out. So that was my first semester, or maybe even semester and a half, of my master's in my education class. Then I picked up. So that's plasticity. So you cannot teach old dog new tricks is false. You could teach an old man new subjects. You can, but it's going to take time. You got to be patient. Am I making sense?
Yes.
It's not like young brainy sponges, right? Your brain's like a sponge when you're young. Mine's like a used dishwasher. It will work, but you got to really sit on it. Okay, so that's plasticity. Okay, so that has to do with age, and that has to do with physiology. So that's plasticity. Is everybody clear about this?
Yes.
And the last principle I want you to know, and these are all organizational principles. So the neurons from the left, right sides of the hemispheres, so left and right hemispheres, okay, cross over. And the scientific term for this is decussate. Okay, which means the right side of my right hemisphere controls the left side muscles. Are we clear?
Yes.
And the left hemisphere controls the right muscles. Is this making sense? They both contain motor neurons, right?
Yes.
This has motor neurons, this has motor neurons, both sides, right? So this one controls this side, this one controls this side. That's called contralateral. Contralateral. Contra means opposite. So this is called contralaterality. Are there any questions on how the cerebrum is organized? Okay. When you go to the subcortical areas, you go into what's called association areas. And when I say the word association areas, what does that remind you? Or what does it tell you? Remember, I smelled vanilla and I had all those responses. I gave that example last class. I did, right? Because
Yes.
I'm singing the same song to another class, right? So I need to know this. So tell me if I mentioned this. So you smell vanilla, you smell you're reminded of your grandmother, or something else. Okay. Anyway, the bottom line, depending on your experience in life, right? It's pleasant or not pleasant. The point I'm trying to make is you need to be able to associate these stimuli, correct?
Mm-hmm.
The sensory and the motor areas are what we call primary areas. They're very, very basic. Yes?
Yes.
They're very basic. It's like a mailman or a mailwoman, right? All they can do is bring letters. They're not going to read it for you and make choices, correct? The board of directors has to make those choices, right? So the board of directors would be your association areas. Are we clear?
Yes.
So you have visual association area. I see this blue water bottle. This water bottle is not mine. It looks hecka ghetto. I ain't gonna touch it. Okay? There, these kinds of associations, right? Or, oh, I know a student who had this bottle. This is hers, right? I should tell her, right? So all of that thanks to association areas. Are we clear? The only thing the primary area does is says that's a blue water. That's blue. That's all it does. It doesn't even say water bottle, right? Even that's association areas. So all processing happens in association areas. Note that down. So all processing. So vanilla grandma connection, that would be your olfactory association area, correct? Olfaction means smell. Okay? So all processing in association areas. You see how my writing is becoming like EEG? It looks so much neater, and then boom, it evolves into this. That means I'm ready to go home. So all processing happens in the association areas. And so there has to be a connection between these things. Don't you agree?
Yes.
Yeah.
So there has to be a connection, and I'm going to draw this in hot pink, right? So there has to be a connection between these neurons and these neurons. Do you agree?
Yes.
Okay? Those are called your interneurons. They're needed for processing. Processing should be rapid or slow. Processing should be rapid or slow.
Slow.
Slow. There's a lion next to you. You don't go, pretty cat. Bad processing. Okay. Do you understand? Processing has to be very careful. You have to be very slow. I'll give you a real life example. You're walking around, you see a tree, and again, we're talking about, good old days when, we were all cave people, right? Yes?
Yeah.
In other words, my high school days. So we were all cave people. You saw a new tree and there's hanging the shiny red fruit, right? There's no way you're going to go grab it and gobble it up. Okay, even animals, what do? I hate saying even animals. Animals are way smarter than we are. What would animals do? They would first smell it.
Yes.
Okay, no dog is going to eat something that you immediately, without even thinking, it's not going to eat it, right?
Yes.
Particularly my dog. They're so intelligent. They love cucumbers more than they like apples, but they have to eat their fruits, and they have to eat their broccoli. I don't want cancer, so I give it to them. So they get fruits and vegetables in the middle of the afternoon. If I give them an apple, my second, dog will absolutely spit it because he smells the cucumbers. But if I don't have the cucumbers out, all I have is the apple. He knows that's all he's getting. It's either that or he gets nothing, right? I send him back to his crate, right? And he hates it. So he'll go, oh, apple, no cucumbers. Okay, I'll eat this. Am I making sense?
Yes.
All that's processing. Processing, of course it's processing. Okay, learning should be followed by processing, correct? So association areas are very important. So interneurons are abundant. And the point is, processing has to be slow, not rapid. Okay?
Yes.
Because processing has to be slow, interneurons are unmyelinated. Note this, I will ask it. Extremely important point. Interneurons are unmyelinated. If you're myelinated, you're like an express train. You will skip stations. Are we clear?
Mm-hmm.
I want you to imagine a motor neuron from my cerebrum to my toe, right? That's 5 foot, 7.5 inches, correct, of a neuron. Do you want the signal to go? Do you?
No.
You have a lion behind you. You don't, right? You want the signal to go rapidly, right? Long neurons are never processing, they're conducting. Long neurons are express trains, okay? The train from San Francisco to New York, by the way, you should do it. It's an amazing journey, 6 days. Did it in 1998. Totally worth it, okay? you think that train's gonna stop at San Leandro?
No.
Oh God, no, right? The next stop to San Francisco is San Jose. It's not gonna stop in all these stations, but BART will.
Yes.
Yes? So BART's your interneuron. Somebody wrote that on the exam, in the essay once. That's an example. BART's not an interneuron. I mean, what is this? But anyway, I'm glad they paid attention once. So the point is, BART stops at every station.
Yes.
If you're a processing neuron, I do want you to do this. Okay? But you'll never see me do this. But if you're an express train, you need to leap. Correct?
Yes.
I ain't leaving. For nobody. Okay? So no leaving. So the point is, long neurons will be myelinated. You need those. Can sodium move through fat? You should know this. That's why sodium doesn't simple diffuse, because it cannot move through fat. Remember, fat in the middle of the membrane. Hello, good morning.
Yes.
Major good morning time. Sodium cannot move through fat. No sodium, no action potential. Good morning.
Yes.
Yes.
That's a good morning squared. Okay, so whenever you have
That's
fat,
good.
sodium can't move through. Are we clear?
Yes.
Yes.
So signals can only happen where there's no fat, right? It's called nodes of Ranvier. So signals jump from one node to another node, and that causes rapid signal transmission. Interneurons have slow signal transmission. Long neurons have rapid signal transmission through their nodes. Are we clear?
Yes.
So I'll leave you at that. We'll continue with that. And we'll also talk about what happens at the end of the neuron, which I forgot to do. So we'll do that next Tuesday. Okay, please remind me. And then we'll wrap up the brain. There's not much left. We'll wrap up the brain, and then we will go into a little bit of spinal cord and then muscle. We'll start muscle. Okay? The lecture exam will only cover up to the end of the nervous system. Are we clear? You have to do sympathetic, parasympathetic, and all that. So we won't do muscle. We'll do autonomic nervous system. Have a wonderful weekend. I will try and post a review video by this weekend. Okay, I will try. Okay. If you have questions, come to office hours.